Good day, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsor, Metabolome, I'd like to welcome you to the translational utility of metabolomics in the integrative omics era. My name is Elizabeth Lamb, and I'm the host and the moderator for today's event. Now, I'd like to introduce our presenter for today. She is Dr. Jessica Ann Lasky-Sue, Associate Professor of Medicine, Brigham and Women's Hospital, and Harvard Medical School. Welcome, Jessica. The presenter ball is yours.
Thank you so much. I am thrilled to be here today, and I'm going to be talking about, as Elizabeth just said, the translational utility of metabolomics in the integrative omics era. And I think this is a topic that is really relevant as we have now entered this omics boom, where we have all sorts of multi-omic data, and research is really trying to combine the data together to discover and understand various disease processes. So I'm going to first give a brief outline of my talk so that you'll know what's coming and you can know how to follow along. I'm first going to introduce sort of metabolomics in the context of how I'm studying it. I'm also going to introduce the cohort that we are going to be discussing and the study objectives of the initial trials for that cohort. And then I'm going to ask some basic questions. Number one is what can metabolomics show us just by itself about individuals and disease processes and how our metabolome looks? We are then going to ask the question, what can metabolomics show us in conjunction with an exposure and a disease outcome? And in this case, the exposure is vitamin D, and the disease outcome is asthma. We're then going to take that same general framework and ask the question, what can metabolomics show us in conjunction with genetics and a disease outcome, where the disease outcome is asthma? We're then going to put all of that together, where we have metabolomics with vitamin D, genetics, and asthma. And so this can really be generalized to a framework of metabolomics, genetics, environmental exposures, and disease. So you can think of it that way if you're interested in other particular diseases. We're then going to add on other evidence from other omics that we have when we actually look at this particular pathway, and what can the other omics that may be available tell us about pathways that we've already identified via metabolomics and genetics. And then the final step is, how can we translate this in a meaningful way into the clinic? So starting out, we know that metabolomics is this global profiling of all the small molecules in a biological system. But I really want us to think about that in the context of what it's telling us, and that is this instantaneous snapshot of a physiological status of a biological system is actually telling us something about an individual's past. You know, whether that past is the preceding omics, like genetics and epigenetics, and environmental conditions earlier in life. It's also telling us a little bit about the present. What is happening right now? Did we just smoke a cigarette? Did we just eat a huge piece of cake? Did we just run a marathon? And what are our disease processes at this moment? Do we have heart disease? Do we not have heart disease? Are we in the middle of an anaphylactic response? But probably in many ways for us and in studying diseases, one of the things that we're most interested in is can this predict future future development of disease? And can this give us some insight into the early stages of disease pathogenesis? So in that context, another thing I want us to consider is this whole concept that metabolites do not exist in isolation. And that is, each individual small metabolite is part of a metabolic pathway that is doing something meaningful. But that metabolic pathway sits within the context of the entire metabolome, which is really monitoring so many aspects and biological systems in our body. But more so, the metabolome is just one part of this omics cascade, where we have all sorts of things influencing it, genetics, transcriptomics, proteomics. And while each one of these omics operates within itself, They also operate in this multi-omic, multi-dimensional way where genetics is influencing the metabolome and the transcriptome, etc. So let's think of this even into the broader context of what I alluded to earlier, which is where all of these omics exist within the context of an environment. And this environment has a lot of influence on several of these omics. And when we're doing this, we're all doing this usually in the context of studying a disease and the development of a disease. So as we look and we think about, how diseases develop, et cetera, there's so much that we need to consider, and it really can be overwhelming. But in the study I'm going to be talking about, really that was one of the goals, is to look and consider not just a metabolite or the metabolome, but the metabolome in the context of multi-omics and the environment, and how can all of those things cause a disease. And in this case, I'm going to be talking about a trial called the VDAART trial, which is an intervention trial of vitamin D during pregnancy. And we follow these kids to look at the risk of asthma later in life. We specifically were hypothesizing that reduced amounts of vitamin D were associated with increased asthma. Put another way, the more vitamin D you give someone during pregnancy, the less the risk of asthma in the offspring. So to go a little bit more into that trial and the design of that trial, this trial was called the Vitamin D Antenatal Asthma Reduction Trial. And women came in very early in pregnancy, 10 to 18 weeks, and they were randomized either to the standard FDA recommended amount of vitamin D, which was 400 IU, or that standard recommendation plus 4,000 additional IUs. And they were given this every single day during pregnancy, from when they enrolled all the way through delivery of the baby. We then had multiple measures of the metabolome, et cetera, from early on through age 3. And then at age 3, we were looking at how many of those individuals were then diagnosed with asthma. And as I alluded to earlier, we used metabolomics. We used the UPLC MFMS approach that Metabolome uses on their standard HD4 platform. And as I'm going to be talking about when it comes to other omic data, we also used genetic information from the OmniExpress chip from Illumina. So why vitamin D? What is the whole story, and what is the rationale behind that? Well, really in the last several years, we have realized that due to a lot of things, and a large part of staying inside and being on computers more and being less active, also in the advent of the big push for sunscreen and protecting us against various types of skin cancer, we have used a lot of sunscreen, and our absorption and now standard levels of vitamin D are substantially less than what they used to be. And in fact, they're at levels that are way below what is recommended. And one of the things that has been hypothesized is that this is a reason that we've had increased incidence of asthma over the last 50 years. And here's some papers, just a very few. When you do Google, you can see thousands of papers on this topic of how lower vitamin D may increase asthma risk and asthma attacks. So in the context of that, we had the VDAART clinical trial, and then our friends in Denmark put on a very similar trial that we will call the TOPSEX trial, and really it was the same study design. And we reported in JAMA in January 2016 these initial findings of the trials, where what we found was, in fact, women who were randomized to high doses of vitamin D during pregnancy had a reduced risk of asthma in their offspring. And the amazing thing is, when you look at this, the reduction in risk was about 23% for both cohorts. So really this protective effect was very, very similar in both groups. Okay, so taking that, and I'm now going to talk about the metabolomic data. So we took this cohort, the VDAART cohort, and we wanted to generate metabolomic profiling in the offspring at age 3. So we used Metabolon, and our first set was a set of 245 samples where we had matched asthma cases and controls, and then we subsequently did another set of HD4 platform with 166 additional samples, giving us both a discovery and a replication cohort within VDAART. And this is all in the context of this much larger trial where we are looking at approximately 1,700 samples from the VDAART cohorts. And at the end of the day, using the 4 platforms from Metabolome with their chemocentric approach that identifies metabolites with authenticated standards, we had 743 named metabolites that we used after both their QC pipelines and our own QC pipelines. Okay, so for the first question we asked is, what can metabolomics tell us just about this cohort in general? And this work was done by my postdoc, Kevin Blythe. So in the initial interrogation of these data, one of the things that we did was this unsupervised clustering of individuals. And one of the things that we noted was there were 3 primary types of offspring that we could identify. And here, when you look to the right, you see these individuals clustered, where you really see this high expression of green here, sort of a neutral in cluster 2, and then a negative in cluster 3, where we see these really profoundly different groups. And then the next question we asked is, well, can we actually characterize what are those metabolites? What seems to be differentiating the 3 groups? And when we did that, what we saw was actually quite striking, and that was that, yes, we could characterize these differences in the 3 groups, and what we saw was this very consistent upregulation of N-6 polyunsaturated fats in cluster 1. And cluster 3 had a reduced level of all these N-6 polyunsaturated fats. Now, to put this into context from biological purposes, N-6 polyunsaturated fat derivatives, metabolites from that, are not the good ones. We like the omega-6s, the N-3 fatty acids. The N-6 fatty acids are associated with inflammatory cascades. They're associated with upregulation of arachidonic acid and all of the subsequent metabolites that lead to inflammation for all sorts of diseases, from heart disease to asthma to many other diseases. So we do not want these guys to be high. And when we look at this, what was actually astounding is when we actually look at the individual metabolites, we just see this very distinct difference in mean levels of these metabolites in the 3 groups. Very consistently, all of these N-6 fatty acids very high in group 1. The other thing we noticed about group 1 was actually associated with the lowest amount of maternal vitamin D during pregnancy. So here what we see, and lowest amount of vitamin D actually in the levels of the children. So here what we see is cluster 1 is this group of low vitamin D supplementation and really high inflammatory markers for metabolites that are, indicative of inflammation. Whereas we see in group 3, there's higher levels of vitamin D, groups 2 and 3, and they are associated with decreased inflammatory metabolites. So at the end of the day, when we ask this question, what can metabolomics tell us about our cohort in general, we see that it actually can cluster the individuals in a very meaningful way, where we can actually see the effects of this intervention and low vitamin D levels associated with these inflammatory metabolites, which biologically, knowing how vitamin D works as a component that reduces inflammation, this actually makes biological sense. So the implication here is that some exposures during pregnancy may actually have a profound and systematic influence on the metabolome. So the next question we ask is, what can metabolomics tell us with respect to asthma and vitamin D intervention? And I just want to point out that this is really the initial intent of the trial, right? How can we see how vitamin D levels actually, during pregnancy, influence the subsequent development of asthma? And this work was done primarily by my postdoc, Rachel Kelly. Putting this pictorially, here we have the vitamin D cohort, the pregnant mothers given the high and low doses of vitamin D. We're really asking the question, can we identify any metabolites that would connect that with increased asthma? So low vitamin D, increased asthma; high vitamin D, decreased asthma. So how would we even go about doing this? Well, the first way we went about doing this is really just asking the question, well, what metabolites are associated with the intervention of vitamin D during pregnancy? And what we saw was there were 26 metabolites associated with this maternal intervention. And can we identify metabolites that are associated with current asthma status? And the answer is yes. We identified 97 metabolites that were associated with current asthma status. But what we really want to know is can we actually identify metabolites that are associated with both, right? We want to make this connection from the intervention to disease. And when we do this, we actually see that, yes, there are 11 metabolites that are common between the two, and actually 3 of them are located in the Sphingolipid pathway, which is of much interest to us, and I will go and discuss why subsequently here. But just know that, okay, so now we've identified these 11 metabolites that are associated with both the intervention and the disease of interest. But if you really think about it, that's not even what we want to get at. What we want to get at is, can we identify metabolites that vitamin D is operating through to cause asthma, right? We want to identify metabolites in the causal pathway to asthma. So how do we do this? We do this with a technique called mediation analysis. And really, mediation analysis is answering this question: can we identify the mechanisms through which vitamin D intervention acts to affect asthma risk by looking at these common metabolites? And by golly, when we do that, what we actually see is, yes, we can. We can actually identify specific metabolites that are driving, that vitamin D seems to be operating through to cause asthma risk. And specifically, these are the 2 groups of metabolites. We have some of these glycerols, and then we also have sphingomyelin. And sphingomyelin was something that really was of particular interest because it's located in the Sphingolipid pathway, and there's many other reasons to believe that that pathway is important for asthma. And I will get onto this a little bit later in the talk. So yes, it actually looks like vitamin D is operating through several of these metabolites to affect asthma risk. That is, increased amounts of vitamin D is actually operating through these pathways to decrease asthma risk. Now, the other interesting thing about this is when we use the same technique with our replication cohort, we also found, in addition to sphingomyelin, 3 other metabolites in the Sphingolipid pathway that were actually showing the same exact association, where the increased vitamin D causes a decrease in asthma via these 3 additional metabolites. So at the end of the day, what is this showing us? Well, we see here's vitamin D in the pregnant mothers. It's operating on this pathway, and we've now identified various metabolites in the Sphingolipid pathway that seem to be important in the development of asthma or the prevention of asthma. So here we see that vitamin D actually causes upregulation of this pathway, because this pathway is good to decrease airway hyperactivity. So I just want us to look at this model, because we're going to be talking about this as we continue on very regularly. The concept here is vitamin D is increasing general sphingolipid synthesis, which decreases airway reactivity. So at the end of the day, when we ask this question, what can metabolomics tell us with respect to asthma and the vitamin D intervention, we can say, number one, there are a good number of metabolites that are associated with both the intervention and asthma at year 3. We actually can identify several of those in the Sphingolipid pathway and also replicate and identify additional metabolites in the Sphingolipid pathway using mediation analysis, which suggests causality. It suggests that vitamin D is operating via these metabolites to reduce asthma. Okay, so now I'm going to add the next layer of complexity to this story, and that is the question. Okay, so we've now seen how vitamin D may operate in conjunction with metabolites to reduce asthma risk. But what about genetics? You know, the beginning of this talk focused in, and I emphasize this whole importance of all these other things operating in the context of disease development. So what can we say about genetics in terms of playing a role in the relationship now between vitamin D, metabolites, and asthma? And that is, when we look at this whole picture that we were interrogating before, we're now adding this genetic component. And what I have drawn here in this genetic component is a very important asthma gene. And that gene is called ORMDL3. And why is this gene so important? Well, when we summarize the last 15 years of asthma genetics, with the era and the advent of genome-wide association studies, the number one most replicated gene for asthma is a gene called ORMDL Sphingolipid Biosynthesis Regulator 3. And here's the initial New England Journal paper that identified this gene in 2010, and subsequently this gene has been replicated multiple times in asthma cohorts as an asthma gene. So why is ORMDL3 so important? Well, I'm showing now in this slide where ORMDL3 sits in the Sphingolipid pathway, right? So if we have vitamin D operating through this pathway, and we also know that this number one very important asthma gene is operating through this pathway, how can we put this all together? So one of the things we know about ORMDL3 through some past research is what ORMDL3 does is if you have the risk variants of ORMDL3, it shuts down this pathway. So you do not want the risk variants of ORMDL3 because it's going to stop this de novo Sphingolipid synthesis and cause airway inflammation. So rather than doing what vitamin D does, where it upregulates this pathway, ORMDL3 is going to stop that from happening. So theoretically, if that's the case, what we should see is with increased copies of the risk allele, we should see decreases in these downstream metabolites. And here's an example of Sphingomyelin, which is one of the metabolites we identified earlier, and the functional variant within ORMDL3. And in fact, what we see is, yes, this decrease in Sphingomyelin levels with increasing number of risk alleles. Okay, so that's talking about the genetics with the metabolomics, but let's now add on the next layer of complexity. So here we just summarize increasing ORMDL3 risk alleles is associated with decreased Sphingomyelin levels. Okay, so here's a picture of that. So ORMDL3 is here. This SPT is what ORMDL3 is inhibiting, and it's actually stopping this whole de novo Sphingolipid synthesis and increasing airway hyperreactivity. So this whole pathway, you put an X through, and it's stopping that and increasing airway hyperreactivity. But now let's look at the next layer of complexity, which is, can we actually link vitamin D to this gene and asthma? And here is a survival plot from the clinical trial. And I want to give you a little context of how to read a survival plot. This starts at when children are born, and this goes throughout life and identifies when these individuals were diagnosed with diseases. So every time individuals were diagnosed from diseases, the line goes down. So the further down this line is, it means more people diagnosed with disease. So here we have the placebo group and the vitamin D group. And then we also have this stratified according to risk genotype. And what we see here is that if you have 2 copies of the risk genotype, it doesn't really matter whether or not you have vitamin D, you're sort of doomed. You're going to get asthma at the same risk, and ORMDL3 operates in full function. However, if you have 0 or 1 copies of the risk allele, what you see is this decrease in risk of asthma. So what it looks like is when you have individuals who don't have the risk allele, vitamin D is able to operate through this pathway and increase the pathway and prevent asthma. But if you have 2 copies of the risk allele, you're sort of doomed, and it doesn't matter how much vitamin D you get, you're probably going to get asthma. Now, I will put the caveat here is maybe if we tripled the dose, maybe actually that risk group would also decrease, but we don't know that yet. So at this point, we see vitamin D has a protective effect on asthma for people with any copy of this wild type allele. And when we see this, one of the things that's amazing is we see the same relationship, not only in the VDAART cohort, but also in the COPSAC cohort, which is our sister trial. And we see this relationship almost identically in both cohorts. Okay, so now let's look at that. So it seems like vitamin D is here. We have ORMDL3 here. And what we see, I'm going to skip to this slide really quickly, is that ORMDL3 stops this production when we have 2 copies of the risk allele, and it doesn't really matter how much vitamin D you get, this pathway is shut down. But if you have any copy of the wild type allele, this does not happen. And one of the interesting things is when we looked at this more carefully in this slide, we actually looked at metabolites in the downstream pathway according to risk genotype. And the only thing I really want you to see here is the GG groups. The GG groups in all these cases are the people with no copies of the risk allele. And for all of these metabolites, you see that those people who have vitamin D, the metabolite is upregulated in this group. You see it's higher here, it's higher here, it's higher here, it's higher here, it's higher here. And while from this plot that may not actually look as statistically significant, a lot of these reach statistical significance. So this is actually showing us, especially that group with no copies of the risk allele, vitamin D is really able to help those individuals and upregulate the whole pathway and decrease asthma. So here's a picture of where those metabolites exist in this pathway. All of those state increase in those who have the risk allele and vitamin D. So as I said before, we now see sort of how this is acting. ORMDL3 shuts down this pathway, but if you don't have the risk allele, vitamin D can operate and push through this, and it doesn't and affects your pathway, upregulate the pathway to cause a decrease in asthma risk. Now, these next 2 slides that I'm going to show you are not meant to intimidate you. I just want to give you this as a perspective of how powerful metabolomics can be in these type of designs. And that is, this is actually all the metabolites that we have data on from Metabolome. And we could actually look at this pathway with respect to a genotype and with respect to asthma and with respect to whether there's vitamin D or no vitamin D. Now, I know that looks really complex, but my point is metabolomics enables us to look at this pathway in great detail while incorporating what we know about genetics and what we know about the environment, and to analyze these data in a very comprehensive way. And actually, in this case, if we were to do all of these metabolites at the same time in this context, what we actually really need is structural equation modeling. And that's actually what our group is currently doing to really examine this pathway at the next level. Okay, so now when we look back and we ask the question, do genetics play a role in the relationship between vitamin D, metabolites, and asthma? The answer is yes. It looks like this ORMDL3 variant is actually associated with decreased levels of many of the sphingolipid metabolites. It also looks like vitamin D can decrease the incidence of asthma in both groups, except those individuals have 2 copies of the risk allele. And we actually are able to replicate those findings quite well. Okay, so I'm now going on to my next level of complexity, and this asks the question, can we add information from other omic data that actually supports our hypotheses? And as you see below, this is actually work from 4 of my collaborators, several postdocs, an assistant professor at Harvard, and associate professor from the COPSEC group who has been visiting us this year. So I want us to go back to the initial picture that we had of this sort of omics cascade with all of these different omics, epigenetics, genetics, transcriptomics, proteomics, metabolomics, and the environment, all to study a disease. And so in this next section, I'm going to give a brief overview of how we actually went about considering some of these other omics and how you can integrate these to examine how they may all be telling a story that is comprehensive and in line with one another. So in the next group of slides, I'm going to discuss these are data that we have on other cohorts that are asthmatic cohorts with other omic data. In the VDAART cohort, we actually have a lot of this data, but at this point, we just haven't had the time to analyze it. So we anticipate we'll be doing similar types of things with VDAART. But I want to show you some work that is either published or in process that actually really can corroborate what we are thinking so far. And the first bit of work is this work that I did with my colleague Mike McGahey here, where he really did probably the first integrative omic approach for looking at asthma that I think has been published using metabolomics as well. And what we did in this analysis was we took epigenetics, genetics, genomics, and metabolomics. And Mike is really an expert in Bayesian Network, so he took all of those types of omics and integrated them together to develop one network of multi-omics and the subsequent development of asthma severity. And he did this using Bayesian networks, and he cross-validated his data, which actually showed he had this really high ability to predict asthma severity. And the very interesting thing about this study is, given all of the metabolites, all of the genetics, all of the genomics, all of the epigenetics, the number one predictor of asthma severity was sphingosine 1-phosphate. And sphingosine 1-phosphate is extremely important and relevant because it's located right in our metabolic pathway. And actually the pathway that seems to be lighting up the most is this pathway right here, and it's really located right in the middle of it. So that was really interesting because we already had that paper published. And so we thought to ourselves, wow, that is actually really interesting. Okay, so the next group of multi-omics I'm going to show you is work that we have done in the context of something called systems biology, which is an approach where we take a lot of an omics data together and generate networks or groups of omics and try to use these networks of omics to describe a disease. So for example, we could generate a network of epigenetics and then apply those networks and see where in that network certain CPG sites are associated with asthma. So I'm going to talk about this approach, and the approach I'm going to talk about, I'm going to give you sort of an overview of the approach just so you get an understanding of what we did, and then I'm going to just know that we actually applied that same approach to epigenetics, transcriptomics, and metabolomics, each individually. The other thing I want to point out is all of the work from here on out was done in another cohort that we have that is a Costa Rican cohort where we have epigenetics, transcriptomics, and metabolomic data. And this is a cohort of asthmatics who are in the age range of 6 to 12, an average age of about 9. So how does this network approach work? Well, the specific network approach we use is something called weighted gene co-expression network analysis. And the idea here is the first step is we take, let's say in this case, it's all of the genetics. And we cluster these genetics using just correlation coefficients. And we can cluster groups of genes that are similar. And from this generation of clusters of similar genes, we can actually take that and draw it pictorially like this and actually identify different groups or what we will call modules of genes. And we have these modules color-coded here. So in this case, we take this, we group them, and then we can either identify really small modules, which is represented by this multicolored up here, or larger groups of the modules, which is identified down here. And that just says these are a group of genes that are acting together. The next step that we do is we take, okay, those groups of genes, and we can ask the question, well, do these groups of genes relate to asthma or asthma traits that are of interest? And in this case, we take this group of genes down here, which is 4 groups, and we say, can we identify a group of genes associated with asthma? And in this case, what we see is when we look at these important asthma traits, we see that, oh, wow, this purple group really has a lot of, it's really associated with a lot of important asthma traits. And then we identify the genes in that module, so all these purple genes, as being likely asthma genes, or actually in this example what we did was all with metabolites, so asthma metabolites. And then we ask the question, well, can we actually characterize those metabolites? Are those metabolites to a specific pathway? Are they telling us something important? And we do this through an enrichment analysis. And in this case, when we did this with metabolomics data, we identify these different modules, and we actually identified that this purple metabolite was associated with sphingolipid metabolism and glycerol-lipid metabolism and glycerophospholipid metabolism. So very interesting. So this network approach is saying, hey, these are the 3 general metabolic pathways that we're identifying that are dysregulated for asthma individuals with all these asthma traits. So we can actually use the same approach for any type of omic data, and that's what we did. And we did this for epigenetics, genomics, metabolomics. And here on the left side, I'm just actually showing you the very, very high-level pictorial summary of what we did. So in this case, here are epigenetics, here are CPG sites, our analysis, the number of modules that we found associated with asthma, all listed here, and the biological processes associated with those modules. So we can do that for epigenetics, and we also can do that for metabolomics. But then this is the key thing. We can then take those modules for metabolomics and those modules for methylation and ask the question, do they overlap? Is there actually an overlap between some of the epigenetic modules and the metabolomic modules? And we can do that and actually identify key modules that are associated with both epigenetics and asthma and metabolomics. And shown in this picture at the very bottom are actually these X's represent modules that are associated together for both methylation and metabolomics. So when we do this sort of integrative approach on a network level with epigenetics and metabolomics, one of the amazing things we find is that the identified immune methylation module with CPG sites, the primary one was at ORMDL3. So when we look at these modules, the number 1 predictor using methylation was actually the CPG sites at ORMDL3. and these were correlated with the asthma sphingolipid metabolomic module. So here we see this evidence on an epigenetic level that we are actually seeing ORMDL3 work, and it's associated with the sphingolipid asthma module. We do the same thing for genomics and metabolomics, and in this case we are only able to identify biological pathways, and we see evidence of increased expression and metabolites for immune signaling, sphingolipid signaling, and phospholipid metabolism. And then we do the same thing for epigenetics and transcriptomics. Now the nice thing about doing this for both epigenetics and transcriptomics is we can now actually narrow things down to specific genes. And when we do that, some of the strongest genes are actually listed below here. And among this list is ORMDL3, gasdermin, which is another gene we know that's associated with asthma, GLB1, and ACR3. So why is it important that those genes are so strongly associated with asthma in both of these epigenetics and transcriptomics? Well, the important thing about that is they're actually located also in the sphingolipid pathway. So now we see this mounting evidence of omic data on many levels implicating the sphingolipid pathway and asthma. So I sort of want to summarize what we have seen so far. In the context of this initial picture I showed where we have all of these omics and we have the environment. So vitamin D was our environment for the VDAART trial. When we looked at the epigenetic data, one of the strongest predictors of asthma was the ORMDL3 CpG site. When we looked at VDAART, we saw that vitamin D actually modifies the effect of ORMDL3 on asthma development. When we look at the transcriptome, we really see sphingolipids implicated in asthma. And then when we look at the metabolome, we see sphingomyelin, S1P, among all the others I showed you in the previous slides. Now, when we look on an integrative omic level, what do we see? Well, we see when we actually look at the epigenome and the transcriptome together, we see 4 relevant genes, 3 of which are in the sphingolipid pathway. When we look at the transcriptome and the metabolome, one of the primary things we see is the sphingolipid pathway. And likewise, when we look at the epigenome and the metabolome, we also see the sphingolipid pathway. So in summary, when you look at this, we see on an individual omic level and on a multi-omic level using network approaches, we see variants in the sphingolipid pathway for genetics and metabolomics implicated in asthma development, and we can identify that vitamin D seems to be modifying these effects. Okay, so when we add on other evidence from other omics, what do we see? We see evidence from epigenetics, genetics, and genomics implicating ORMDL3, gasdermin, and 2 other genes in the sphingolipid pathway, sphingomyelin, S1P, many other metabolites in asthma pathogenesis, evidence that vitamin D alters these relationships. So I want to go the next step, and this next step is really just taking this to the clinic. So what, we now really see that it really does look like there's compelling evidence on multiple levels that the sphingolipid pathway is involved in asthma pathogenesis. But how can we really translate this to the clinic? Because really that's what we're looking for, right? How can this become something we can utilize in a medical setting? And one of the first things that we've done, and our group has actually done, this is not a paper published by our group, but our group is actually doing this now, is you can actually develop this ORMDL3 knockout mouse. And one of the most interesting things about the knockout mouse is the most difficult aspect of doing studies is that this mouse has a lot of trouble with breathing because of all the increased airway remodeling and airway hyperresponsiveness that the mouse has. So certainly implicating that ORMDL3 is important in asthma. The other thing that is very interesting is when we actually look at this pathway, and here's another example of just looking at the pathway where here you have ORMDL3, it's inhibiting SPT, and the rest of the pathway is being downregulated. That when we actually look at this pathway and we look at ORMDL3, we actually now have already seen literature that has identified sphingosine 1-phosphate, which is the rate-limiting step in the whole sphingolipid pathway. As you know, sphingosine 1-phosphate is formed by the sphingosine kinases that have been identified as possible drug targets in the treatment of asthma. And actually, sphingosine 1-phosphate is already being used to treat other autoimmune conditions. So really suggesting that some of the metabolites in these pathways may be good targets for future clinical research. So in conclusion, what have we learned at this point? Well, number one, we've learned that the metabolite can differentiate clusters of people that may be related to important exposures and or disease. And one of the key things we saw was the striking relationship between vitamin D intervention and the upregulation of all of these bad N-6 metabolite derivatives. Metabolomics also enables us to identify mechanisms through which exposures and genetics may impact disease. And we show this by really identifying how we could look at some exposure, and in our case it was vitamin D, and decompose how that may be operating with a gene and metabolites to cause asthma. And then we can also validate these findings by adding evidence from other omic data to support our findings. And really, I truly believe that I think metabolomics is the omic that is really going to help us bring the multiple billions of dollars that have been spent on genetics and genomics and epigenetics and bring that to actually be clinically relevant. So that's the end of my talk. I just want to acknowledge these four people at the bottom, Bo Chow's, Kevin Blythe, Mike McGahee, and Rachel Kelly for all of the hard work they've done on this. Also Scott Weiss and Gus Latwanwa, who are the PIs of VDAART, and of course my funding sources and Metabolome, who really has been a tremendous support of these efforts. I really have to say the amount of work they have done behind the scenes to really help us, especially when we had multiple runs of things on the HD4 platform, the tireless work that they have done to help us merge that data together. I cannot, it really was a concierge type service that they've provided for us in a very expedient way. So I just want to thank them for all their hard work as well. So that ends my talk, and I'm happy to answer any questions.
Thank you very much, Jessica. We do have a few questions for you. Our first question, were vitamin D exposure levels measured in the trial?
Yes, that's a great question. So vitamin D exposure levels were actually measured in the mothers and the children, and they continue to be monitored. And these relationships I saw, so actually the relationship I actually showed in the slides of the maternal vitamin D level and the subsequent increased risk of all these inflammatory metabolites, that was actually the actual vitamin D levels in the mothers. So regardless of whether we actually look at actual vitamin D levels or the intervention, we see the same thing. And in fact, when we look at the actual vitamin D levels, these associations seem stronger. In general, just because of the context of a clinical trial and the integrity of maintaining that study design, that's why we often look at intervention instead of the actual levels, but the levels support what we see.
Okay, perfect. Our next question, have you looked at a correlation with RSV infection in an infant related to these studies?
A great question. So yes, RSV is something that in the VDAART trial and our sister cohort on the COPSAC trial, we have examined. And one of the things I do want to point out and say is this is one mechanism by which I think, the vitamin D may be protective in asthma, but it's certainly not the only environmental risk factor that can cause asthma exposure. We do see increased incidence of asthma with RSV, and that I think is really a pretty established correlation, and I totally believe it. Yes, so it's a great question. Yes, we do see that relationship.
All right. Our next question, what tissue did you use for the generation of the epigenetic data?
So epigenetic data, we used peripheral blood, and the primary reasons for that is, honestly, that cohort, when it was sampled, that's what we had available, and so that's what we used. I will say what we did the transcriptomics on and the epigenetics on was the same blood, the same time points, and the same time point that we also evaluated the various asthma disease phenotypes.
Okay. Are there any urinary metabolites associated with asthma which have been proved in clinical trials?
So that is a fantastic question. Whoever asked that question should get a gold star, because some of the work I have done, I've been talking to, and some of the people I've collaborated with really have suggested that looking at specifically the arachidonic acid pathway is probably best seen in urine. And the other advantage of urine is it's so easily obtainable, and it sort of really can represent what's happening right at the moment. So there has been work done. Craig Wheelock is the person who's spearheading those efforts. He's a colleague and a collaborator of mine, but he has actually convinced me that if I'm doing studies in the future, especially of asthma exacerbations, and I want to look at the arachidonic acid pathway, that urine is probably the best way to go. Unfortunately, in infants and unpotty trained kids, it can be really difficult to get urine because it's hard to time it.
Okay. And another attendee asks, what was the composition of the vitamin D formula used in the intervention? Did it contain other fats?
No, it was just a supplement of vitamin D, and it was the 25-hydroxyl vitamin D. That's sort of the standard one that is administered as a vitamin.
Okay, so would that be D3?
Yes.
Okay. What are some good tools to integrate metabolomics data with other omic data?
So I think one of the things we did here, and I'm really convinced, I think this is probably the best way at this point to integrate omics data, is to look and see what clues you have about what metabolic pathways may be involved. And, gathering that from genetics, gathering that from what you know about maybe environmental exposures or what you know about the disease physiology to begin with. Like, for example, we know that asthma develops, one of the ways it develops is through, increased leukotrienes, which is part of the arachidonic acid pathway, and we use leukotriene inhibitors. So we know that biology. So I think knowing that biology can help you focus and target your work on a pathway. And then I think it really takes well thought out study designs that actually obtain biospecimens in real time to then really take it to the next step where we can actually see what's happening. So, potentially getting the biospecimens during an exacerbation or something like that. Now, if you want to know the way, let's say, okay, well, I have these genes, and I even have some disease, some medicines I know that treat my disease, and some metabolites that I think are involved, but I don't really understand it all. There's some online tools that you can actually use to see what the literature says up to this point on relationships between pharmaceutical drugs, diseases, genes, and metabolites. And one of the best tools I would recommend is a website called Stitch that is really useful, and it generates networks that you can look at. They're sort of like a cytoscape type network, but that we found useful.
Okay. I know you mentioned that you used blood for the epigenetic tissue. Did you also use blood for the metabolome analysis?
Yes. In this case, and I'm sorry I didn't mention that, everything was blood and epigenetics and metabolomics were all done on the same blood at the same time. And, that's where really the question of what biospecimen is the optimal one to use really comes in, and that's why I say I think urine may be a good one and be easily obtainable. We have talked about things like exhaled breath condensate, or probably the best one would be bronchoscopies. Of course, that's sort of an invasive. There's also some concerns about some of the things with exhaled breath condensates, but that's also something I'm interested in looking into further.
Okay, looks like we have time for just one more question. What's your view on the utility of this multi-omics approach to responder analysis for drugs targeting central nervous system diseases?
So I am not necessarily an expert in central nervous system diseases, but I do think, if you are able to, and I have talked to various colleagues and various researchers looking at diseases where CSF would be the biospecimen you'd want to use. I think it's potentially tremendous. I really think in that case it comes down to, I think getting the biospecimen, which would be CSF. So an example where I think this could be helpful would be like a disease where, let's say something like Alzheimer's disease, where you have certain genes that are influencing it, like ApoE. That's a very strong indicator of Alzheimer's, using that in conjunction with metabolomics, and we also know it's a brain disease, right? So that, the other good thing about something like that is you can just narrow down a lot of the metabolic pathways you're looking at to ones that are important in brain function. And so for that, a good one would be sort of the tryptophan serotonin pathway is sort of an obvious choice when it comes to some of these, diseases of the brain. And that is something that you can do quite easily with metabolomics. So narrowing your search down to biological pathways that are relevant for the brain, and then, the good thing about a lot of these neurological diseases is there are genes that have been identified and validated, like I said, Alzheimer's, schizophrenia. Some of these diseases really are further along in terms of gene identification, which makes the multi-omic integration even more ideal.
All right. Excellent timing. I see we've come up on the hour. I'd like to thank you so much, Jessica, for your presentation today. I'd also like to thank Metabolon for sponsoring today's web symposium. And most of all, I'd like to thank those of you who came and spent some time with us. Hopefully you gained some information that will make your research a bit easier, and we're very grateful you spent this time with us. We look forward to seeing you at future CHI Global Web Symposia. Thank you, and have a great day.